Welcome to the deep dive. Today, we're plunging into, well, a world you might not think about much, but it's absolutely teeming with incredible life and surprising impacts.
That's right. We're talking fungi.
But not just, mushrooms on your pizza. We're going much deeper into a fascinating group known as the Pyrenomycetes.
Exactly. These are fungi defined by some really unique features in their reproduction. Think microscopic sacs, called asci, often tucked away inside tiny flask-shaped bodies.
Flask-shaped. Okay.
Or sometimes completely enclosed ones. They're a major part of a larger class, the Hymenoascomycetes.
So our mission today is to explore these guys, their structures, their life cycles, which are pretty clever, their roles out there in nature, and their significance for us. Medicine, farming, even history.
And we'll try to paint a picture with words, since, well, you can't see these microscopic details directly.
Right, relying on the sheer coolness of their biology.
Okay, so to start, what really sets Pyrenomycetes apart is how their asci, those little spore sacs, are built, and how they work.
You mentioned asci.
Right. They're usually what we call non-fisitunicate, or protocinicate.
No.
Fancy words. But it basically means the sac walls don't split open along a predefined line.
Ah, okay. So how did the spores get out?
Yeah, either through a little pore, or sometimes the whole sac wall just kind of dissolves. It's more of a controlled release, not like a popping balloon.
Gotcha. And these sacs are inside things.
Usually, yes. Inside structures called perithecial, those are the flask-shaped ones, or sometimes totally sealed ones, called cleistothecia.
And you mentioned they belong to the Hymenoascomycetes class. What does that tell us?
That tells us how the whole fruiting body, the ascoma, develops. It forms in what's called an ascohymenial way.
Meaning
The structure itself forms after some key cellular events happen, like cells fusing and their nuclei pairing up. It's all very organized. The asci then arise from a fertile layer inside, called a hymenium.
Okay, so it's not just random spore sacs floating around.
Not at all. It's quite different from other fungal groups where asci might be more scattered.
So picture this perithecium, this flask. What's going on inside?
Well, it forms around an initial cell, the ascogonium. Then these fertile threads, the ascogenous hyphae, start growing inside. A wall develops around it all.
Okay.
And you might also find sterile filaments inside, called periphyses, growing up from the base, and others called periphyses, lining the opening.
And that opening, the ostiole, how's that made? You said it's like a valve.
Right. It typically forms through a schizogenous process. The tissue literally pushes itself apart to create the opening.
Yeah.
It doesn't dissolve. It splits open precisely.
Wow. Nature's engineering at a tiny scale.
Absolutely. And then spore release. It can be active, using water pressure, turgor pressure, like a tiny water cannon, or passive, where they just ooze out in a sticky mass, sometimes colotendril or cirrus.
Okay, structure's fascinating. But what do they do? What are their lifestyles like?
Incredibly varied. Many are saprotrophs, the recyclers, breaking down dead stuff in soil, on wood. Huge ecological role there.
Essential decomposers.
Definitely. Others live with plants. Some are mutualistic endophytes, living inside plants and actually helping them. Others, though, are pathogens, causing diseases.
And some even go after animals.
Yes, particularly insects. Some are specialized insect parasites.
How can they do so many different things?
A big part of it is their chemistry. They produce a huge range of biologically active metabolites. Things like alkaloids, antibiotics, plant toxins, powerful stuff.
Powerful and potentially dangerous, as you hinted.
Potentially, yes. As we'll see, these chemicals can have massive impacts.
Okay, let's zoom in a bit. You mentioned different groups. How about the Sorderiales?
Ah, yes. A really important order. Think genera like Sorderia, Podospora, and the famous Neurospora.
And their niches. Interesting.
Often, yes. Many are coprophilous. They love dung, herbivore dung specifically.
Growing on poop. Okay, but why are they important?
Because these humble dung fungi, especially Sorderia fimicola, became workhorses for geneticists. Their peritheci have this neat little sphincter-like mechanism at the opening for releasing spores.
Right, the controlled release.
And Sorderia dispersal is amazing. It shoots out projectiles with spores inside. It can be one spore, can be up to 8.
And you mentioned something counterintuitive about distance.
Exactly. You'd think a single spore would fly furthest, right? Less weight.
Yeah, seems logical.
But no. The projectiles with more spores travel further. It's about aerodynamics. The surface-to-volume ratio means the bigger clumps cut through air resistance better.
Wow. Okay, physics and fungi. And they aim these things.
They do. The necks of the peritheci are phototropic.
Yeah.
They grow towards light, so they shoot the spores away from the dung pile, hopefully onto fresh grass where an animal might eat them.
Clever strategy.
And the spores themselves are tough. They have this sticky mucilage envelope to cling to plants. They can survive drying out, even develop tiny gas vacuoles to help them float.
And passing through an animal helps them.
It actually enhances germination for many. The gut passage primes them. Perfect adaptation for that lifestyle.
So this precision, the shooting, the colors, that's why they're good for genetics.
Precisely. Even though Sorderia fimicola can self-fertilize its homothalic, you can cross strains. Say one makes black spores, one makes white.
Okay.
In the resulting asci, those sacs, you often see a perfect 4 black spores and 4 white spores. The pattern tells you how genes are linked and positioned on chromosomes. It's visual genetics.
Like reading a genetic map right there in the fungus.
Exactly. But sometimes you get weird results like 5 black and 3 white.
Uh-oh. What's happening there?
That's gene conversion. One version of the gene, one allele, actually converts the other one. It's not a simple swap. It's like one rewrites the other during the process of DNA recombination and repair.
Oh!
Yeah. Understanding this in fungi helps scientists develop models like the holiday model, explaining how DNA strands can cross over and exchange information. It gave us fundamental insights into DNA mechanics.
Amazing what you learned from dung fungi. Okay, what about Podospora and Serena? You mentioned that one too.
Right. Podospora shows us something called pseudohomotholism. It looks self-fertile because a single spore can start a new reproductive cycle.
But it's not truly self-fertile.
Genetically, no. It's heterothalic, meaning it still needs 2 different mating types. The trick is that its spores usually package nuclei of both mating types inside that single spore.
How does it manage that?
Through really complex, controlled nuclear movement inside the developing spore, guided by the cell's internal skeleton, microfilaments, and microtubules. It ensures genetic mixing while simplifying dispersal.
Very efficient. And Podospora also shows us incompatibility.
Yes, heterogenic incompatibility. If 2 Podospora colonies that are genetically different try to fuse their hyphae, their cells often react badly.
Like rejecting a transplant.
Kind of. It's an antagonistic reaction. Sometimes you see a clear line, a barrage, where they meet and kill each other's cells off.
Why do that?
It's a defense.
Ugh.
It stops potentially harmful things like fungal viruses, mycoviruses, from spreading from one colony to another through those fused cells.
Makes sense. Protect the individual colony.
But here's where Podospora gets really mind-bending. There's a specific genetic system, the Hetchet-S system. It controls one type of incompatibility, and it functions like a prion.
Wait, like prion diseases, like mad cow disease, in a fungus.
Functionally, yes. There's a protein, Het S, that can misfold. And once it misfolds, it causes other Het's proteins to misfold too, forming aggregates. That triggers the incompatibility reaction. It's an infectious protein conformation, just like a mammalian prion.
That is staggering. Finding that mechanism in a fungus.
It really shook things up, showed these fundamental protein misfolding processes might be more widespread than we thought. And Podospora is also a key model for studying aging, or senescence.
How so?
Well, colonies in the lab have a limited lifespan. They age and die. And you can actually transmit this senescence factor from an old colony to a young one just by letting their hyphae fuse.
You can infect it with old age?
Sort of. It provides a way to study the biological basis of aging in a relatively simple, fast-growing organism.
Incredible. Okay, one more from this group. Neurospora, the genetic superstar.
Neurospora crassa. Absolutely fundamental. It was used in the studies that led to the one gene, one enzyme hypothesis, the idea that each gene codes for one specific enzyme. Nobel Prize-winning work.
A foundational concept in biology.
And its genome was one of the first fungal genomes fully sequenced. In nature, you find it on burnt ground.
Why burnt ground?
If spores are dormant in the soil, and the heat from a fire is the trigger that makes them germinate, they're pioneers after a fire. Though it can also be a pest. It grows fast and produces masses of orange spores. It can quickly overwhelm things in bakeries, for example, nicknamed red bread mold.
Right, but it's a big contribution beyond basic genetics.
Circadian rhythms, our biological clocks.
It helped us understand our clocks.
Massively. Research on Neurospora identified key genes and proteins involved in regulating daily cycles. The same fundamental mechanisms operate in practically all eukaryotes, including us. Understanding how this fungus keeps time helped unlock how we keep time.
Wow. From bread mold to biological clocks. Okay, let's shift gears. From genetics to, well, fungi doing damage and good in forests and fields. The Zellerialis.
Yes, often found on wood. You might see their stromata, these hard, often black, carbon-like structures on branches or logs.
What are they doing there?
Many are powerful wood decomposers. They cause white rot, breaking down the tough lignin in wood. But some are also significant plant pathogens.
And they have sneaky strategies.
Some do, like latent invasion. Take Hypoxylon fragiform, common on beech trees. It can live inside the wood of a perfectly healthy tree as tiny hidden inoculum units.
Just waiting.
Exactly. Kept in check by the high moisture content of the living wood. But if the branch dies or the wood starts to dry out,
Then it takes over.
it spreads rapidly, a real opportunist strategy.
And you mentioned dramatic spore release earlier. Does this group do that?
Oh, yes. Hypoxylon fragiform has this amazing spore release called eclosion, a term usually used for insects hatching.
Eclosion. What happens?
When the spores sense specific chemical signals from the host tree, things like monolignyl glucosides, they react incredibly fast.
How fast?
Within milliseconds, the outer spore wall bursts. Then, over about 10 seconds, the inner part expands dramatically and the spore pops out. It's like a microscopic jack-in-the-box triggered by the tree itself.
A highly specific host recognition system. Amazing. Okay, let's move to the hypocoreales. Brighter colors, you said.
Often, yes. Brightly colored perithecial. And a huge diversity of asexual forms. They're anamorphs.
What roles do they play?
Again, very diverse. Soil and water saprotrophs, major plant pathogens, and also mycoparasites.
Fungi eating other fungi.
Yes. Some attack cultivated mushrooms, for instance. But their biggest impact on us comes from their chemistry. They are metabolic powerhouses.
Producing lots of compound.
A staggering array of secondary metabolites, things not essential for basic growth, but used for defense, communication, competition.
Like what?
Potent antifungal compounds, like gliotoxin and viridin, to fight off rivals. Crucial antibiotics, cephalosporin, came from a related fungus.
Cephalosporin. Huge in medicine.
Absolutely. And, perhaps surprisingly, plant growth hormones,
Yeah.
gibberellins.
They make plant hormones.
They do. It was discovered because one species, Gibberella, causes foolish seedling disease in rice. The infected seedlings grow ridiculously tall and spindly.
So a disease symptom led to finding a useful growth promoter.
Exactly. That hormone is now used widely in agriculture. And there are other benefits.
Such as?
Well, the meat substitute corn. That's made from the processed mycelium of a fusarium species, which belongs to this group.
I did not know that.
And another fungus, Trichoderma resi, is an industrial workhorse for producing cellulase enzymes.
Cellulose, breaking down plant fibers.
Right. Used in making biofuels, processing textiles, even in food production.
Okay, lots of benefits. But you mentioned a darker side, fusarium.
Yes, the Nectriaceae family, especially fusarium. Many species are devastating plant pathogens. They cause wilts, cankers, root rots in countless crops. Huge economic damage.
And they affect humans, too.
Unfortunately, yes. Some fusarium species can cause human infections. Anything from nail or eye infections to really serious, life-threatening systemic infections, especially in people with weakened immune systems.
And they're hard to treat.
They can be. They're known for developing resistance to antifungal drugs relatively quickly.
But the biggest threat might be toxins.
Mycotoxins, yes. Fusarium species are notorious producers. Things like zearalenone, which messes with livestock reproduction, causing infertility,
Oh, wow.
and a group called trichothecines. Includes T2 toxin, vomitoxin.
Yeah.
These can make animals and humans seriously ill, damage the immune system by reducing white blood cell counts. They contaminate grain crops worldwide.
A hidden danger in our food?
Which brings us to one of the most infamous examples.
Ergotism.
St. Anthony's fire, that's the one. Caused by eating rye or other grains contaminated with Claviceps purpurea. This fungus has a complex life cycle involving producing the sugary honeydew liquid on infected grain heads to attract insects, which then spread its spores.
And if people ate that contaminated grain?
The results were horrific. Two main forms. Gangrenous ergotism, where blood flow to extremities was cut off, leading to limbs literally rotting and falling off.
Horrified.
And convulsive ergotism, causing seizures, muscle spasms, terrifying hallucinations, mania, psychosis, plus an intense burning sensation.
Right.
Hence, St. Anthony's fire.
People died from this.
Oh, yes. Entire villages could be affected. There are theories, quite plausible ones, that outbreaks of ergotism with the hallucinations and strange behavior may have fueled some of the witchcraft accusations in places like Salem.
Wow. Fungi potentially influencing historical events like witch trials.
It's a stark reminder of how potent these fungal chemicals can be.
Ah.
But there's a twist.
A good twist.
Yes, those same toxic ergot alkaloids from claviceps became the source of vital medicines.
Oh.
Compounds were isolated, like ergometrine, used to control bleeding after childbirth by causing uterine contractions, and ergotamine, used to treat migraines.
So poison becomes medicine.
Exactly. And maybe the most famous derivative, Lysergic acid, another ergot alkaloid, was synthesized into lysergic acid diethylamide.
LSD.
LSD came directly from research on ergot alkaloids, a substance with a complex history, from medicine to counterculture.
It all ties back to this fungus on rye. Incredible. So how do we fight back against the bad guys, like fusarium causing wilts?
A lot of effort goes into breeding resistant plants, resistant host cultivars, finding plants with natural defenses,
Makes sense?
and there's also biological control, using other harmless fusarium strains that can outcompete the pathogenic ones in the soil, protecting the plant.
Fighting fungi with fungi. Let's keep going. What about the clavicipitels? You mentioned cordyceps earlier, the insect killers.
That's right. Many in this order are specialized grass pathogens or live harmlessly inside grasses as endophytes. But cordyceps is famous for parasitizing insects.
The zombie ant fungus and things like that.
Exactly. They infect the insect, take over its body, consume it from within, and then fungus erupts, growing its fruiting body right out of the insect's corpse. Fungal mummies.
Gruesome but fascinating.
Some, like Cordyceps sinensis, found on caterpillars in the Himalayas, are incredibly valuable in traditional Chinese medicine.
And there's an evolutionary leap here.
A really cool one. Some cordyceps that attack underground truffles, which are themselves fungi, are very closely related genetically to species that attack cicada nymphs, which live underground.
So it jumped from attacking an insect to attacking a fungus, or vice versa.
The evidence suggests an interkingdom host jump, maybe from insect to fungus, perhaps because both live underground in similar conditions. It's a rare evolutionary event.
Life finds a way. And medicine from this group, too.
A huge one. Tulipocallidium inflatum, which is the asexual stage of a cordyceps-like fungus, produces cyclosporin A.
Cyclosporin. That's the anti-rejection drug for organ transplants, right?
The very same, a cornerstone of transplant medicine, discovered from an insect parasitizing fungus.
Mind-blowing. Okay, shifting again. Ophiostomatales. Sounds ominous.
It includes the fungi responsible for Dutch elm disease, caused by Ophiostoma species.
Which wiped out so many elm trees.
Devastatingly so. And it's not just the fungus, it's a disease complex. The fungus relies on specific bark beetles to carry its spores from tree to tree.
Vector insects.
Right. The beetle tunnels under the bark introduces the fungus, which then gets into the tree's water transport system, the xylem vessels.
And clogs them up.
Yes, the fungus grows, and the tree also reacts defensively, producing blockages called tyloses. Either way, water flow stops, branches wilt, and the tree eventually dies.
But there's a counter story here, something about viruses.
Yes, hypovirulence. A really interesting biological control angle. Scientists found some strains of the Dutch elm fungus were much weaker, less virulent,
Why?
because they were infected themselves. Infected by fungal viruses called B factors made a double-stranded RNA.
So the fungus got sick.
Essentially, yes. The virus messes up the fungus's growth, its ability to make spores, even makes it sterile sometimes. It weakens the pathogen.
Can we use that?
People have tried. Introducing these hypovirulent, virus-infected strains into diseased trees, the hope is the virus spreads to the aggressive strains, weakening them. It's like using a virus to treat a fungal disease.
Viral therapy for trees. Okay, next up. Magnaporthaceae. Rice blast disease.
Magnaporthecrecia. Yes, arguably one of the most destructive plant diseases on the planet. A huge threat to rice production, a staple food for billions.
How does it infect the rice plant? It must be effective.
Incredibly effective. Its spores land on a leaf and form a specialized infection structure, and a pressorium.
The pressorium.
Yeah.
Sounds like it presses.
It does. But how it presses is astonishing. It builds up enormous internal water pressure, turgor pressure,
How much pressure?
up to 8 megapascals. That's 80 bars, or nearly 1,200 pounds per square inch.
1,200 psi in a single fungal cell.
Yes. Generated by breaking down stored fats and pumping in solutes like glycerol to draw water in osmotically. It's one of the highest pressures recorded in any living cell.
What does it do with all that pressure?
It focuses it onto a single point and pushes a tiny rigid penetration peg right through the tough outer layer, the cuticle, of the rice leaf. It literally punches its way inside.
A biomechanical marvel, just brute force penetration.
It really is. Understanding the signaling pathways inside the fungus that control this, things like the CAN-AP pathway, MAP kinase pathways, is key research.
Can we target those pathways to stop the disease?
That's the tricky part. Those signaling pathways are fundamental. They're highly conserved, meaning very similar pathways exist in other fungi plants, even us.
Ah. So a chemical that blocks it in the fungus might also harm the plant, or beneficial insects, or even be toxic to humans.
Exactly. Finding fungicides that target these core pathways specifically without collateral damage is very difficult. You need more unique targets.
Right. Okay, one last example. Hymenoascomycetes graminis. Take all disease.
Yes, in wheat and other cereals. A root-infecting fungus that lives in the soil can cause massive yield losses, over 50% sometimes.
Sounds grim.
It can be,
Hmm.
but there's a fascinating natural phenomenon called take-all decline.
The disease just declines.
Yes. After a field has had severe take-all for several years, often if you keep planting wheat there, the disease severity naturally drops off. The soil becomes suppressive.
Why? What changes in the soil?
The soil microbial community changes. Specifically, populations of certain bacteria like fluorescent Pseudomonas species build up.
And they fight the fungus.
They do. They produce antifungal compounds like one called 2-volafor-diacetyl-fluoroglucanol that inhibit Hymenoascomycetes. The soil microbiome essentially develops its own immunity to the pathogen.
Nature's own biological control kicking in. Hashtag, tag, outro. Wow. What an incredible journey through the world of Pyrenomycetes. It's just staggering. From, the basic building blocks and genetic tricks like gene conversion.
To those prion-like proteins, which is just amazing.
Right. And then the insect zombies, the life-saving drugs like cyclosporin, the devastating diseases like Dutch elm or rice blast, the historical terror of ergotism.
It shows their incredible adaptability. Decomposers, partners, pathogens, chemists, they do it all.
They really are shaping ecosystems and impacting our lives in ways we rarely see or appreciate. Powerful stuff from microscopic organisms.
Constantly reminding us of that intricate web of life, much of it hidden from view.
So as you listen to this, what stands out? Maybe it's the sheer ingenuity, like that high-pressure punch of Magnaporth, or the surprising connections fungi influencing witch trials or giving us transplant drugs.
Or perhaps just the realization that understanding these complex fungi is key. Key to harnessing their benefits, like new medicines or biocontrol agents, and key to managing the threats they pose to our health and food security.
There's clearly so much more to learn. Thank you for taking this deep dive with us today.
Always a pleasure to explore the hidden kingdoms.